Friends, welcome back in another video tutorial from microbiology with Chakrik. So in that video tutorial we are going to discuss about a recombinant DNA technology application. Although in our previous video we have discussed about the site directed mutagenesis, which is also a recombinant DNA technology application where we are modifying protein at single amino acid. And today we are going to discuss about how with the help of recombinant DNA technology we can produce insulin. Okay, so first thing is that the insulin which is produced with the help of recombinant DNA technology are commonly known as humulin. Okay, so this humulin was first discovered by David Godel and its colleagues of Gentech of USA, and later it was marketed by Lilly under the name humulin. So what is the utility of humulin? Humulin is used to treat diabetes. Okay, and here we are producing humulin inside a bacterium. That means we are using the bacterial system to produce the insulin. Okay, so insulin produced by recombinant DNA technology are known as humulin, and these humulin are synthesized by recombinant DNA technology, and it was first discovered by David Godel and its colleagues of Gentech of USA, and later it was marketed by Lilly under the trade name humulin. Okay, so here we are incorporating the gene of insulin inside a bacterium, and we are using the bacterial system as a biofactory to produce insulin. So let's move to the next slide. So the first question is, what are the advantages of recombinant insulin? Why we use recombinant insulin instead of using another methods? So previously, before recombinant insulin, we are using animals insulin. That because the insulin hormone which regulates the blood sugar level or regulate the sugar level in blood is also present in cow, also present in pigs, but there can be allergic reaction, or there can be transmission of infection in such cases. Okay, and we have to faces various kind of ethical issues too, because here we have to kill the animal and then isolate the insulin from them. So there are some ethical issues too. So recombinant DNA technology help us to bypass all of that problems. First of all, no risk of transferring infection, no allergic reaction compared to cow and pig insulin, and no ethical issues concerning slaughtering and the use of animals. And the most important thing is, it allow us to produce large quantities of insulin in short time. Okay, so first of all, as we are artificially synthesizing insulin, that means that the structure of insulin is very clear to us, right? Otherwise we cannot use any kind of recombinant DNA technology tool to produce our desired product, right? We have to know what we are going to produce first, right? So let's discuss about the structure of insulin a little bit. So structure of insulin was first discovered by Frederick Sanger. Okay, and he showed us that insulin is consist of two ring or two chain. One is A chain, another one is B chain, consisting of 21 and 30 amino acid respectively. Okay, and this A chain and B chain are joined by disulfide bond. Insulin is comparatively the simplest protein present in human. That is why we are capable of producing insulin. Okay, and more specifically, insulin is the first approved drug produced with the help of recombinant DNA technology. Okay, so let's revise what we have learned up to that part. So insulin is a hormone that regulates the glucose level in blood. Okay, so now the problem is we have to produce insulin artificially in large amount to fight against diabetes. In previous time we are using the insulin of other animals. Okay, but there are chance of allergic reaction, transmission of infection, and there are some ethical issues too. So recombinant DNA technology, after the development of recombinant DNA technology, scientists thought that they can make insulin artificially with the help of RDT. Okay, as at that time Friedrich Sanger already described the entire structure of insulin. That entire structure of insulin is consist of one A chain and B chain, and A chain and B chain consisting of 20 to 30 amino acid respectively, and this A chain and B chain are joined together by disulfide bond. So we thought that we can construct all of these things using recombinant DNA technology, and David Gödel and his colleagues in the laboratory of Gentech in USA first discovered the insulin, or I should say first developed the humulin, and later Lilly marketed it under trade name humulin. And don't forget that it is the first approved drug produced with the help of recombinant DNA technology. Now we are going to see the structure of the insulin. Not only that, we are going to now enter what are the steps that we have to follow one by one to produce humulin. So let's discuss. Okay, now this is a schematic diagram of human insulin. So as you can see, it is a chain A and consists of two chains, chain A and chain B. Chain A consists of 21 amino acid and chain B consists of 30 amino acid. Okay, chain A, chain B, and this chain A and chain B are joined together by disulfide bond. That is the important thing. Okay, so Friedrich Sanger is first provide the structure of insulin. Now let's see what are the different type of process that we need to follow one by one to produce our insulin. Okay, so let's think that what should be the process that we want to follow one by one to produce insulin. So first of all, we need the gene, okay, so that we can clone them and amplify them in vivo. So our first target is to isolate gene, and before isolate we have to identify, right? So we can isolate our gene of interest, that is here the insulin producing gene, from cDNA. Okay, why? Because cDNA lacks intron, so it could be very useful to us. Not only that, we can isolate the gene from tissue, okay, and then after isolation of the gene we can use PCR amplification to increase their number. Okay, after the PCR amplification we can also use very chemical synthesis method to synthesize them chemically. Okay, and the last one is the most preferable one. So we can chemically synthesize it, which is the most preferable one. We can also isolate it. Okay, for isolation we have to isolate the gene from the tissue. Okay, and after the isolation we can simply perform PCR technique to amplify them, or we can use cDNA library, okay, of human to find out the gene, insulin gene containing cDNA clones, right, or cDNA colonies. Now, so after the isolation and identification and isolation of gene, our next goal will be the construction of the vector. Okay, and remember one thing, there will be two vector. Okay, because here we are synthesizing both a insulin A gene and insulin B gene in two separate culture. Okay, and later we will join them with the help of disulfide bond. Okay, so we need two vector, although the construction of both the vector will exactly be the same. So first thing that we need is a promoter inside the vector, because remember here we are going to produce insulin protein, right? That means we need the expression of the gene, so a promoter is necessary. Along with that we will incorporate beta gal. Okay, so this beta gal will induce transcription of gene and also ensures that our protein of interest is also transcribing. Okay, and not only that, this beta gal will be incorporated along with the insulin A chain, so that it will lead to the formation of fusion protein, that is a complex of beta gal and insulin A chain, so that this beta gal will help us to separate our insulin A protein and insulin B protein with the help of affinity chromatography. Okay, So that is why we have to incorporate beta gal just beside the insulin A chain. Okay, along with we will also incorporate a selectable marker, okay, for primary selection, that means for selection of the transform cell, and we can also include here a secondary marker if we want, and the most importantly the origin of replication for autonomous replication, right, because we cannot rely on our cell to replicate, so we have to wait for the one cell division, right, because they replicate once in a cell division. Similar way we will also produce the insulin B chain containing recombinant vector. Okay, so after the construction of the vector, we will transform them with standard transformation technique. If we are using E. coli as a host, then we can perform electroporation. Okay, we can perform liposomal mediated gene delivery. I have described all the electroporation and liposomal mediated gene delivery in detail in my various videos, right? So if you want to know what is electroporation, what is liposomal mediated gene delivery, what is microinjection, you can simply find those videos in my YouTube channel and watch. Okay, so remember one thing, we will prefer electroporation or liposomal mediated gene delivery whenever we are using E. coli. Okay, but if our preferable choice is Saccharomyces cerevisiae, because don't forget here our insulin is a eukaryotic gene. Okay, so we should prefer Saccharomyces cerevisiae, which is yeast, which is a E. coli cell, which is a eukaryotic cell for production of yeast, production of insulin, sorry for that. So because this should be helpful to us, although we can also use our prokaryotic gene, that is prokaryotic cell, that is E. coli too. So after transformation of the E. coli cell, our next challenge will be to isolate them, right? So for, isolate our recombinant DNA containing or recombinant vector containing plasmid, recombinant vector containing E. coli. Okay, so how can we screen our recombinant DNA containing or recombinant vector containing E. coli? So that is why we have incorporated antibiotic resistant gene. Okay, not only that, we can look assay for various biological processes. We can use colony hybridization, we can use various blotting techniques. Okay, we can isolate the DNA from the colonies and then we will screen them. We will what we will do, we will sequence them to find out whether our gene of interest is present here or not, and we can also isolate our gene and simply apply them in primer gene specific PCR. Okay, where we are developing a primer that will find out whether insulin encoding gene is successfully incorporated or not. Okay, so after those kind of screening procedure, we will only pick up those colonies that giving us positive result. Okay, after that we will subculture them, okay, in large scale. So after the subculture, our next goal will be the expression. Okay, so let's focus on that vector. As we have here incorporated promoter, if I use lactose in medium, this lactose will induce promoter to transcribe. Okay, so this transcription phenomenon will also ensure that our protein of interest is also transcribing. Okay, so after transcription and translation, the beta gal and insulin A chain, and along also in another case the beta gal and insulin B chain will fuse together and leads to the formation of fusion protein. Okay, and due to the presence of beta gal, we can easily separate beta gal containing insulin chains from the rest of the protein. Okay, because beta gal containing insulin A chain or beta gal containing insulin B chain can be easily separated with the help of affinity chromatography. Okay, so after separation, we will upon separation we will simply add cyanogen bromide. Okay, this cyanogen bromide or CNBr will break the methionine, okay, within the insulin A chain and insulin B chain. Okay, so then we have our either purified A and B chain, and we will join them with the help of disulfide bond under appropriate condition. So after the formation of disulfide bond between purified A and purified B chain, we have our active insulin. Okay, so that is the entire procedure by which insulin can be produced. So our first goal will be the identification and isolation of the gene from which we can produce insulin. So we can isolate our gene from the tissue and can amplify with the help of PCR technique. We can chemically synthesize them, which is the most preferable one, and we can also use them and we can also isolate them from the cDNA library. Okay, so after the isolation, we have now our gene of interest. So our next goal will be is the construction of the vector. The vector should contain the promoter along with the beta gal, and beside the beta gal we should have insulin A chain. Okay, so we will use two vector, one for insulin A chain production and insulin B chain production, another one into separate culture. Okay, along with those important things, a primary selection marker along with origin of replication, both are necessary for the production of insulin A chain, okay, and insulin B chain. So next is the screening of recombinant vector containing cell. Okay, so here we will use antibiotic that we have incorporated. If we do not want to use antibiotic, we can use the morphology, okay, of colony. Not only that, we can also use the assay that they show for various biological properties, and most easiest technique that we have discussed also earlier, that is colony hybridization. Okay, the technique that we use to screen libraries. So colony hybridization, we can also use blotting technique. We can isolate the DNA from the colonies and sequence them to find out whether the colony contain our gene of interest or not. We can also isolate the DNA and apply PCR amplification with specific primer, which will find out which is designed specifically complementary for the insulin gene. Okay, that is the various kind of screening procedure that we can use to find out our transformed E. coli. Okay, once we have find out our transformed E. coli, we will simply subculture them, and after the subculture we will go for the expression, right? So we will add lactose in the medium. This lactose will induce the transcription of beta gal along with the insulin A chain, and in another case the insulin B chain. After the transcription, it will translate and give us fusion protein, that is a fusion of beta gal and insulin A chain in case number 1, and in case number 2 the fusion of beta gal and insulin B chain. This fusion partner beta gal in both the chain will help us to isolate insulin A and insulin B chain from rest of the proteins. Okay, so once we have isolated insulin A chain and beta gal and insulin B chain and beta gal into separate container, we will use CNBR. This CNBR will break the methionine within the insulin A chain and separate insulin A chain and beta gal. So now we have purified insulin A and insulin B, and we will simply join them with disulfide bond. Okay, this disulfide bond should be used, they can be joined with appropriate temperature and appropriate conditions. Okay, so after the joining of the formation of disulfide bond between insulin A and insulin B, we have our functional insulin. Okay, so that is the procedure that we have to follow to form insulin, that is recombinant DNA technology based insulin or humidin. So let's discuss what we have learned today. Okay, so we are here, our main goal here is to produce insulin with the help of recombinant DNA technology, right? So it was first discovered by David Gödel and his colleagues of Gentech USA, and later it was marketed by Lilly with the trade name Humulin. Okay, and Humulin is the first drug produced by recombinant DNA technology that is approved. Okay, and now, so what is the need of producing insulin with the help of recombinant DNA technology? So the answer of that question is, remember, in case of before the development of insulin with the help of recombinant DNA technology, so what is the need of producing insulin with the help of recombinant DNA technology? So earlier, before the development of recombinant DNA technology, we are simply using the animal insulin, right? And for that purpose, we have to kill them. So there are some ethical issues, and not only that, the animal insulin may be allergic to someone, and there is also a potential chance of transferring infections, right? So as the remarkable development of recombinant DNA technology, and Friedrich Sanger explained us the detailed structure of insulin, that it is consists of two chain, one is chain A and chain B. Chain A consists of 21 amino acid and chain B consists of 30 amino acid, and these two chain are joined by disulfide bond. So now we can scientists try to produce insulin with the help of recombinant DNA technology. So David Gödel and his colleagues succeeded in that experiment, and they first developed recombinant DNA technology synthesized insulin. Okay, so that steps are, first we have to isolate, before isolation we have to identify the gene of interest. Okay, so gene of interest can be identified by identified and isolated from the cDNA library. It can be chemically synthesized, which is the most preferable one. We can isolate our gene of interest, and once the isolation, we will amplify them with the help of PCR. Okay, not only that, then we will develop the vector. Don't forget that here we will going to use the expression vector. So there will be a promoter along with beta gal and insulin A chain. Okay, and antibiotic resistant gene, and also ORI, that is origin of replication. Okay, so we have to use two vectors for the synthesis of insulin A chain and B chain separately into different culture. Then we will transform them in E. coli or Saccharomyces cerevisiae with the help of electroporation, microinjection, or liposomal mediated gene delivery. So after the transformation, the next goal will be the screening or selection of our recombinant vector containing host. So how can we perform this? We can perform this simply by using the antibiotic that we have incorporated. Okay, not only the use of antibiotic, we can also use colony hybridization, we can use our another blotting technique. We can isolate the DNA and sequence them with the help of standard sequencing method that we have. We can also isolate our gene of interest, and then we can also isolate the genes from the colonies. Okay, and then we will use PCR amplification. Here we are providing the primer that is specifically complementary to the insulin gene. Okay, that is how we can find out which colonies contains the recombinant vector containing gene of interest. Okay, then we will simply select those colonies and subculture them in large scale. Okay, after the formation of those cells, as we are using expression vector, we have to insert lactose in the medium. This lactose will induce the transcription of gene. Okay, not only that, along with it will also ensure that our protein of interest is also transcribing, right? So after transcription and translation, we will have fusion protein, that is beta gal along with insulin A chain and beta gal along with insulin B chain. This beta gal will provide us to isolate the insulin A chain and insulin B chain protein from other proteins with the help of affinity chromatography. After purification, we will add CNBR. Okay, this CNBR will break the methionine bond and separates the beta gal from the insulin chains. Okay, so now we have purified A and B chain. Now we will form the disulfide bond between purified A and B chain under appropriate condition, and in that way we can generate a active insulin, okay, with the help of recombinant DNA technology. So that is for today, guys. I hope that this video will be helpful to you. If it is, please hit the like button, share with your friends, and subscribe to my channel to get more videos like that.